Yes, sir.
Evelina, get ready. Like that.
Yes, sir.
A very good morning to all the participants of this historic, distinguished oration program. Professor Shuviresh Bhattacharya, the Vice Chancellor of North Bengal University, the members of the organizing committee of international conference, the faculty, the researchers, the students from North Bengal University, and also from GIS Institute of Advanced Studies and Research, large number of thousands of participants who are viewing this program from YouTube or from Facebook, and my dear students.
Dr. Ray, please, for interruption. Dr. Ray, first there will
Mhm.
be information, after that you start, please.
Okay,
Okay? Yeah,
okay.
yeah, yeah. For the time being, please wait. Invocation will start first, after that.
Oh,
You give
acha.
your Debolina will request you. Debolina will request.
Thank you,
Debolina, please
sir.
start.
A very good morning to all the dignitaries present here, along with the research scholars and the students. I welcome you all to the International Conference on Confluence of Chemistry and Biology, Exploring New Frontiers, jointly organized by Department of Chemistry, University of North Bengal, JIS Institute of Advanced Studies and Research, Kolkata, Chemical Research Society of India, North Bengal Local Chapter, West Bengal. In association with Department of Chemistry, CMS College, Kerala, and Department of Chemistry and Center for Research, St. Teresa College, Kerala. It will be our privilege to have with us our Honorable Nobel Laureate, Dr. Ada E. Turner. Now, with all your permission, may I now request Monishita Nondi, research scholar of University of North Bengal, to kindly start our convocation with her inaugural song. Please, Monishita Nondi ji.
Thank you, Debolina. And at first, welcome and a very good morning to all of you. And I want to convey my deep regards and respect to the honorable chief guests and all of my respected professors and teachers to express my honor through my music, which I am going to perform now. It's not only a music with some combination of rhythm, tune, pitch, etc., but it's the music of soul, music of our nation, and obviously the music of entire universe too. It's an worship and devotion of ourselves to the ultimate power, the mother of this universe. And we all know her in our Bengali language as Jivan Devata, the god of our internal soul. This mesmerizing concept of Jivan Devata and the magical creation of musical worship to the god inside the flesh and blood was drawn really flawlessly by none other than our Nobel laureate, our guru, our strength, our inspiration, Sri Rabindranath Tagore. I just want to build a bridge of admiration in between the renowned Nobel laureates present here, Professor Yonath ma'am, and the magical tune of the another Nobel laureate of our nation, Sri Rabindranath Tagore. त्र्यम्बकं यजामहे सुगन्धिं पुष्टिवर्धनम् उर्वारुकमिव बन्धनम् मृत्युर्मुक्षीय ममृतत् ॐ মা গামাদানি রা গামাদানি রা রে না রে না রে না রে না না শক্তি আদি পরম জল জ্যোতি তোমারি গগনে গগনে প্রথম আদি তব শক্তি তোমার আদিবাণী বহিছে তব আনন্দ তোমার আদিবাণী বহিছে তব আনন্দ জাগিছে নব নব রসে হৃদয়ে মনে প্রথম আদি তব শক্তি আদি পরম জল জ্যোতি গগনে হৃদয় ভাতে সূরজ চন্দ্র তারা প্রাণ তরঙ্গ উঠে পবনে তোমার চিদাকাশে ভাতে সূরজ চন্দ্র তারা প্রাণ তরঙ্গ উঠে পবনে তুমি আদি কবি কবি গুরু তুমি তুমি আদি কবি কবি গুরু তুমি মন্ত্র তোমার মন্ত্রিত সব ভুবনে প্রথম আদি তব শক্তি আদি পরম চল জ্যোতি তোমারি হে গগনে গগনে প্রথম আদি তব শক্তি। Thank you so much.
Thank you, Monishita Di, for your beautiful song. May I now humbly request for Padma Shri Professor Dr. Ajay Kumar Ray, Director, JIS Institute of Advanced Studies and Research, Kolkata, to kindly deliver the welcome address on behalf of the organizing committee. Please, sir.
A very good morning to all the participants of this great conference, which is being organized by North Bengal University, the JIS Institute of Advanced Studies and Research, in association with CMS College, Kottayam, Kerala, St. Teresa's College, Ernakulam, Kerala, and CRSI North Bengal local chapter. So this is a great occasion for all of us to extend a very, very warm welcome to our guest, distinguished speaker, Professor Ada Yonath, who received his Nobel Prize in Chemistry from Weizmann Institute of Science in Israel. Professor Yonath has kindly consented to deliver a distinguished oration on next generation antibiotics to this vast audience who are enjoying and who I'm sure will be immensely inspired by her talk on how to do research and then about the recent advances in the field of research in not only ribosome, but also on some of the new directions of new generation antibiotics. Professor Yonath received her Nobel Prize for her outstanding contribution in the area of understanding the ribosome's molecular structure using X-ray crystallography. So she started doing his research almost now about 4 or 5 decades back. And during this whole period, she has, like a rishi, as Monishita had talked about, she has really done a very, very important aspect of understanding its structure. Ribosomes, as are all well known, they are protein building structures, which is at the core of all living cells. Professor Ionath's work has also led to the understanding of how antibiotics work. It is very interesting that even after 10 years, 11 years of her receiving the Nobel Prize, she is still one of the most prolific scientists in this area in the whole world. She is the director of the Center of Biomolecular Structure and Assembly at the Weizmann Institute of Science in Israel. She is still continuing her work in search of newer antibiotics. Her contribution to the mankind, to the welfare of the mankind, especially in this grim scenario of the pandemic caused by COVID-19, will always be remembered by the entire humanity. In fact, not only as an Asian, but also as one of the very small entities in the whole universe, very small entity of life, we all are greatly indebted to the brilliant work that Professor Ada Yonath has done and is still pursuing. So I'm sure that with her fantastic research mind, I'm sure that the whole world will get the next generation of antibiotics, which will be a great thing for the whole world. So I once again extend a very warm welcome to Professor Ada Yonath. And at the same time, I wish that the youngsters who are attending this, thousands of them who are attending this program, they will be very much inspired by her, this great, wise oration. So I would like to take advantage of uh, extending my thanks to Professor Subiresh Bhattacharya, who is present here as the Vice Chancellor of North Bengal University. And I'm sure that jointly, all our researchers in these two institutes, JIS Institute of Advanced Studies and Research, is possibly the youngest research institute in the entire country. We started hardly 1 year back, but then we are trying to strive very hard to really achieve excellence and in understanding the diverse aspects of science in interdisciplinary areas. With these few words, I once again thank Professor Ada Yonath for accepting our invitation and for agreeing to deliver today's great oration. Thank you very much.
Thank you so much, sir, for your kind words. May I now request Professor Anirban Mishra, Head, Department of Chemistry, University of North Bengal, and also the Chairman of the Organizing Committee to kindly deliver some words. Please, sir.
Very good morning to you all. Greetings from the Department of Chemistry, University of North Bengal. Honorable Vice Chancellor of our university, Dr. Subhiresh Bhattacharya, Professor Ajay Kumar Rai of GIS Institute of Advanced Research, today's illustrious speaker, Nobel laureate Professor Ada Yonath, my dear colleagues from the University of North Bengal and other institutes, all the dignitaries present in this auspicious occasion, research scholars, and my dear beloved students. It is my proud privilege to welcome you all in this today's international seminar on Confluence of Chemistry and Biology, Exploring New Frontiers, organized jointly by the Department of Chemistry, University of North Bengal and JIS Institute of Advanced Studies, Kolkata, Advanced Research, Kolkata, and Chemical Research Society of India, North Bengal Local Chapter, in association with the CMS College, Kottayam, Kerala, and St. Teresa's College, Ernakulam, Kerala. Today, Professor Yonath will talk about next generation antibiotics. The importance of this discussion is obvious in this unprecedented situation of the present global pandemic. I am sure that all of us present here will be immensely benefited from today's deliberations. I personally thank all the organizers of today's event, especially to our senior faculty member, Professor Pranav Ghosh, who is also the convener of CRSI North Bengal local chapter, for making this event possible. Last but not the least, I would like to mention that our Department of Chemistry, University of North Bengal, and Professor Yonath have a very close connection, as one of our brilliant students, Dr. Tanaya Bose, is presently working in Professor Yonath's group as a postdoctoral research associate. Once again, I thank you very much for joining us, and I am sure that next 1 hour will be memorable to all of us forever. Thank you very much.
Thank you so much, sir, for your words. May I now humbly request our Honorable Vice Chancellor, Dr. Subhiresh Bhattacharya, University of North Bengal, to kindly deliver the inaugural address. Please, sir.
Very good morning to everyone, wherever you are. This is indeed a great pleasure for me to be here with you all in this international conference jointly organized by North Bengal University, JS Institute of Advanced Studies and other colleges of the country. Respected Professor Ada, respected Dr. Ajay Kumar Ray, Director JS Institute of Advanced Studies and Research, Dr. Kakuli Bose, Scientist, Tata Memorial Center for Cancer Research, Professor Mishra, Head of the Department of Chemistry, NBU, other faculty members of the department, my beloved research scholar, students, and other participants, and the convener of the conference, the architect of the meet, Professor Pranav Kumar Ghosh. Chemistry is often called the central science because of its role in connecting chemistry with the life science and applied sciences such as medicine and engineering. The nature of this relationship is one of the main topics in the philosophy of chemistry and in scientometrics. Thus, chemistry is built on an understanding of laws of physics that govern particles such as atoms, protons, neutrons, electrons, thermodynamics, etc. Although it has been shown that it has not been fully reduced to quantum mechanics. Concept of such the periodicity of the element and chemical bonds in chemistry are emergent in that way they are more than the underlying forces defined by physics. In the same way, biology cannot be fully reduced to chemistry, although the machinery that is responsible for life is composed of molecules. For instance, the machinery of evolution may be described in terms of chemistry by the understanding that it is a mutation in the order of genetic base pairs in DNA of an organism. However, chemistry cannot fully describe the process since it does not contain concepts such as natural selection that are responsible for driving evolution. Chemistry is fundamental to biology since it provides a methodology for studying and understanding the molecules that compose cells. Connections made by chemistry are formed through various subdisciplines that utilize concepts from multiple scientific disciplines. Chemistry and physics are both needed in the areas of physical chemistry, nuclear chemistry, and theoretical chemistry. Chemistry and biology intersect in the areas of biochemistry, medicinal chemistry, molecular biology, chemical biology, molecular genetics, and immunochemistry. Chemistry and the earth sciences intersect in areas like geochemistry and hydrology. It is felt that chemistry is changing its face to a great extent due to its overlap in one side with mathematics and physics and on the other side with biology. I therefore appreciate the Department of Chemistry for choosing the theme of international seminar, the confluence of chemistry and biology, exploring new frontiers. This overlap on both extremes has reduced chemistry to lose its original color, but it now exhibits a variety of colors like a rainbow. In fact, demarcation line that existed between chemistry and biology is slowly vanishing. This changeover is going to have many classifications in the study of chemistry. This shift in emphasis will have many other consequences. Some of the changes that one can expect in the study of chemistry would be that the study of atoms and molecules and clusters will become routine and mapping of the wave functions will become possible, and hence chemical reactivity will become a predictable parameter. The consequence of the overlap of chemistry with biology will manifest remarkably in our study of the energy conversion process. I will not take much of your time because Professor Ella is there to deliver her lecture on next generation antibiotics. I'm sure the participants who are attending this international seminar will be immensely benefited by the deliberation of the resource persons. I'll request the participants, please get enriched out of the deliberation made by the resource persons and disseminate this experience to those who are not attending this seminar. I wish this international seminar a grand success. Thank you. Thank you very much.
Thank you so much, sir. Now, moving forward, we are going to start our today's session. This session will be chaired by Dr. Kakoli Bose. It is a great privilege to introduce our eminent speaker, Dr. Kakoli Bose. She is the Scientific Officer and Principal Investigator, ACTREC, Tata Memorial Center, Mumbai. She is also the head of Integrated Biophysics and Structural Biology Group. Her current research is focused on elucidating protein-protein interactions involved in the apoptotic pathways using multidisciplinary tools that include structural biology, biophysics, and cell biology. She has completed her master's in chemistry from University of Calcutta and PhD from Biochemistry Department of North Carolina State University, USA, and her postdoctoral research from Boston, USA. She has received the National Women Bioscientist Award from Government of India and the sorry, beg my pardon, and I would like to request warmly Dr. Kakuli Bose to kindly chair the session. Please, ma'am.
Thank you so much, Debolina, for your kind words. Very good morning to all. First, I would like to wholeheartedly thank the organizers, especially Professor Ghosh, for inviting me to chair the session. It will be an honor to be a part of this event as Dr. Yonath has a special place in my heart, be it for being a chemist who's working in the field of structural biology, or be the first Israeli woman to receive Nobel in chemistry. But above all, she inspired me with her endurance, perseverance, focus, and grit. She walked towards her goal silently for several decades that initially seemed impossible to the world, and the rest is history. Before we start the lecture, I would like to share a few words about Professor Ada Yona. She was born in Jerusalem. Professor Yonah later moved to Israel with her family, working hard toward continuing her education. While in school and college, she graduated from the Hebrew University of Jerusalem with a bachelor's degree and then master's degree in biochemistry in 1964. In 1968, she obtained her PhD from Weizmann Institute for X-ray crystallographic studies on collagen. Her brief postdoctoral research in Carnegie Mellon University and MIT she did till 1970. During her postdoctoral tenure in MIT, she spent some time in Harvard as well, working in the lab of subsequent 1976 chemistry Nobel laureate Professor William N. Lipscomb, where she developed the passion of solving crystal structures of huge biomolecules. Later, she returned to Weizmann and built the first protein crystallography unit there. Simultaneously, she headed Max Planck Institute research unit in Germany between 1986 and 2004. A great discovery is always a brainchild of a curious mind. Let me share an interesting anecdote in this context. In late 1970s, Dr. Ada met with a serious bicycle accident and had to stay away from work for months. She spent this time judiciously reading about polar bears. In spring, when they come out of hibernation to start metabolism, their ribosomes might be arranged in an orderly fashion. This gave her the idea that there is a way to closely pack the ribosome. To pursue this challenging goal, she worked with thermo and high salt resistant bacteria from Black Sea and found a way to preserve their activity and integrity during crystallization. She started working on ribosomal structure in 1979, and her publications on the final structures came out in 2000 and 2001. So, for 2 decades, she worked with immense focus and determination towards solving the structure and understanding the intricate functions of ribosome. Her hard work led to the path-breaking discovery that ribosome is a ribozyme that strategically aligns its substrates for peptide bond formation and subsequent catalysis. For ribosomal crystallography possible, she developed a novel technique called cryobiocrystallography, which has opened the floodgate for scientific discovery to the structural biologists worldwide that otherwise almost seemed unimaginable. Professor Yonath received innumerable accolades during her glorious scientific career, to name a few. In 2002, Israel Prize and Harvey Prize. In 2006, the Emmett Prize for Art, Science, and Culture. Then she received Albert Einstein Award of Science. In 2009, it was a proud moment for Israel when she received the Nobel in Chemistry that she shared with her peers Thomas Stites and Venkatraman Ramakrishnan. She later received Marie Curie Medal from the Polish Chemical Society. In 2020, she was elected a foreign member of the Royal Society. Many of us might not know she was also the first Israeli woman scientist to work with NASA in their 13 missions and to send research material to outer space. Professor Yonath has not only been an amazing mentor to the scientists, students, and postdoctoral fellows who have crossed her path, but have also been a beacon of inspiration for the entire scientific fraternity. Now, without further ado, I invite Professor Ada Yonath and request her to deliver her talk, for which we all are eagerly waiting. Thank you very much, and I would kindly request Professor Ada to deliver her speech. I think she's having a problem logging in. Can you please help her? Dr. Yonath is having problem with joining in and she's being logged off, I think. She put up a message over here. So before that, I would like to tell, since she is having problem and others might be working, so after her talk, there will be 10 or 15 minutes of question answer session. Once you can type your name and serially, I can ask you to ask your questions to Professor Yonath, because here we don't have the option of raising hands. So whoever wants to ask questions at the end of the talk can type your name through the message option, and I can ask your name and you can please ask your question.
Yeah. Good morning, Dr. Bose. Yeah, you are right. Dr. Ada Yonath has some problem in linking with us. So she has recorded her lecture and she has given the link. So we will go for the recorded lecture to listen right now.
Okay, so will there be any question answer or there won't be any question answer then?
Let's see, she is still trying. If we can get in, then we'll continue with this. Okay.
Sure.
So now listen the particular lecture in recorded version.
Good day.
Okay.
I would have liked to be here today with you, but since I cannot, at least I sent my lecture and I would like to tell you about what is it. Good day. The translation of the genetic code into its products, the proteins, and how one can stop it if one wants to stop the life of a pathogen by antibiotics. Antibiotics are fantastic, but there are problems with them. The main problem is resistance, and we are trying now to fight against it, and that's about what the lecture talks. Thank you. I want to talk about next generation antibiotics, or at least what we suggest should be next generation antibiotics. And for this, I also want to show my motivation, which was actually curiosity, like that of our hero, Mr. Einstein, that said that all what he had was curiosity and passion to it. When I became a young scientist, I thought the translation of the instructions embedded in the genetic code into proteins is one of the most interesting life process. Therefore, I focused on it. What do I mean? Let's first see what proteins do. And what I use here is some pictures from a children's book about proteins in human. But actually proteins perform nearly all the essential functions in every living cell, even in bacteria, in flowers, in trees, and in human. So here there are some proteins. Hair and skin, structural proteins, signaling proteins like insulin, transport proteins like hemoglobin, sensory proteins like vision, and enzymes that are taking apart the proteins that we eat to their components in order to make new proteins, those that we need. So what we know about proteins, some of them perform almost all cellular functions, as you saw previously, even muscle motion, and some are responding to pressure, like heat or cold shock, sometimes heart events, and so on. Proteins are long chains made of amino acids. There are 20 types of amino acids. The fold of each protein, which is carefully designed for fulfilling its function, is determined by the sequence of the amino acids that are building it. So what I want to tell is in nature, structure means function. Look at this machine, very sophisticated machine that is called paperclip. It works only when the fold is correct. where the structure is correct. If it's being modified by fingers, without heating, without cooling, without cutting, without anything, just changing this structure or the fold, there is no function. And this is the way proteins work. So as I said earlier, there are 20 types of amino acids, and their sequence within the proteins, which enable its functionality, is dictated by the gene that codes for proteins. So proteins are made according to their genes, which are in the DNA. Actually, the DNA is a string of instructions written in a 4-letter language in which each 3 letters, or we call them also bases, are coding for a specific amino acid. These are called codons, these 3 letters, these triplets. So this is a typical enzyme. It's made of many atoms, many amino acids, each made of atoms. It has a active site. And if we look into the active site, this is here, and this is supported by the structure around it. I rotated it and I enlarged it, and you can see the active site now with the substrate. And you see that the substrate fills it exactly. If it was too large, it won't fit. If it was too small, it will flip around. So it must fit this exact. So this must be exactly according to what nature does. In this case, what nature needs is this for this substrate. So how do the proteins, are they made? Here there are 5 amino acids, 5 components. All of them have, it's part of the 20, yeah? All of them have the same backbone, and all of them have the backbone that can fit each other, and each of them has a different side chain that gives them the biological or the chemical character, and some of the side chains can also be biological tools. They get together, they become more and more and more together, so long that I didn't even draw the details. Then they become long protein. About each normal proteins are about between 200 to 500 amino acids, and it's worth nothing unless it is folded with an active site, as you could see earlier in the real protein. So this was known when I started my work, but it was not known how it happens. This is what I wanted to find out, how this happens. So many other people wanted to do it. They were more experienced and they had better labs, and they failed. So there was a big skepticism about the possibility to understand it. There were even papers saying it cannot be done. Yet, I tried to do this. I will tell you afterwards why. I knew that we also can fail, but I knew that if we don't fail, if we make it, we will have a big smiley. And this is what we have today. So the central dogma says the DNA that you can see here is where the bases are. It means the letters, the triplets that code for the proteins. The information is inside here. These are the bases, these are the letters within the double helical structure. And here it is not available for reading. It's covered, it's hidden. So the first thing that happens, there is a transcription, to a very similar molecule that can exist as a single chain. So here the letters are exposed and can be used. They are available for using. And this can be translated to growing proteins by a particle within every cell that is called ribosomes. Actually, there are many ribosomes within every living cell. So they are universal. It means they function in a very similar way in all cells, regardless of the source. It can be bacteria, it can be flower, it can be cockroach, it can be human. They do the same. The decoding and the production of the protein the same way. There is a huge number of ribosomes that function in every living cell. Even in bacteria, they can reach 100,000 when they work. And in some mammalian cells can contain millions. For instance, the liver may have 5 to 6 million ribosomes. In vivo, the ribosomes act continuously. They can form up to 40 peptide bonds in a second. They hardly make mistake. So I, as a very good and fast student, needed 6 hours for one bond, but they can do 40 in a second with about mistake rate of about 1 to a million. So now that we know how they work, we like to describe them as a factory that we call ribosomes that is made of 2 floors. In the top floor, the information is being decoded. The triplets are coming in. It means the whole messenger RNA comes in, and triplets, when a triplets come here, it's found what it is coding for. The amino acid that is cognate to it will come inside into the factory and be connected to the growing protein that goes out. This arrow had to be inside, but I don't know how to draw it, so I drew it outside, but it happens in the factory. Then this track will become empty and join the other empty tracks. And as I told you, there are 2 floors. Here the decoding is being made, and here the making of the protein, the making of the bond, which is called peptide bond. So actually what you saw in the factory is shown here now in molecular way. Ribosomes, all ribosomes are made of 2 subunits, small and large. These 2 subunits are independent in the cell. They live as small and large. They come together when ribosome has to work. And they are connected by contacts between them and by the track, the tRNA molecule that brought the amino acids. So here it is, and it connects between the decoding center and the peptide bond formation center, which is called PTC. So this is what we found after 20 years minus 2 weeks of work. And we made a little movie that is available in YouTube that you can see here. Here, messenger RNA comes to the small subunit that waits. There are factors in the cell that are coding that are telling the ribosomes that has to start to work. There are initiation factors. In bacteria, there are 3 of them, and what you see here is actually ribosome from bacteria. And the whole thing is monitored by them. So yes, there are many more factors. You will see some in a minute. So messenger is reaching the small subunit that takes it. We made the movie, us together with 2 students of the Art Academy in Jerusalem. And the messenger now will go into its path in the small subunits, the top floor of the factory. And it's monitored by initiation factor 3. When this is okay, initiation factor 2 is bringing it the first track, the tRNA molecule with the amino acid, this one. And now where everything is okay, the factors go out and the large subunit can come and associate with the small one by their surface and by making bridges. Now the ribosome is ready to work. We look at it from the solvent side. Up is the large subunit, below is the small one. And you can see elongation factors bringing it inside the tracks. The tracks are being decoded and peptide bond is being made. And you can see the ribosome helps. And it makes very fast, 40 of them in a second. So you can see here how the decoding happens in the small subunit. And from the large subunit, we left only the tunnel through which the protein goes out. So this is how it is being protected when it's made. Now you can see the whole ribosome again. The tunnel is inside there in the large subunit. And the process continues until the protein is large enough. Here it is being grown and coming out. And when there is a stop codon, a codon that says stop, then instead of tRNA comes a factor, recycling factor and release factor. These factors separate between the small and the large subunit. The protein can fold and come out and has to do what it has. And the tRNAs, the tracks, and the messenger and the ribosome are waiting for the next job. That's all. That's the whole story. So what we saw in the movie was rather fuzzy, but what actually happens is that we know the position of each and every atom in the ribosome, small subunit, which sediments as 30S and large subunit is 50S. You can see that the ribosome is made of many components. The main one is ribosomal RNA, another RNA molecule, and many ribosomal proteins in many colors here. And just to remind you, the small subunit is doing the decoding, and the large is peptide bond, is for peptide bond formation. Now let's look at these people. All of them are giants, either in music, like Mozart, Chopin, Schubert, or very intellectual writers. And what's common to all of them is they died very young. You can see their lifespan. The oldest is 47. Also, what's common to all of them, they died before or at the middle of last century. What you cannot see in the picture, but I can tell you, is they all died from infectious diseases that people don't die now from them. So what happened in the middle of last century? In the middle of last century, antibiotics came into use. And they saved the lives of many, many people, and of course, extending lifespan. So because of the fundamental role played by ribosomes, many antibiotics target them. Almost half, over 40% of the clinically useful antibiotics target protein biosynthesis, mostly by paralyzing the ribosome. What are the natural antibiotics? They are the weapons that bacteria from one type use to interfere with the life of other species of microorganisms when they have fights for space or for food or so on. Question is, how do the tiny antibiotic paralyze the giant ribosome? So bacterial ribosome have a molecular weight of 2.5 million Dalton, and antibiotics less than 1,000. And the trick is that they are bound to the active sites. So you see here the small subunit and the large subunit, and the circles are showing where are families of antibiotics. It means the natural antibiotics that were modified by companies. And you see all of them are bound to functional sites, either to the decoding, or to the bonding, or to the tunneling, or to the hinging. These are the motions that are needed for continuing. So this is the trick, and we want to show you a movie that was made based on the previous movie, and it's already 13 years ago, and as I told you, on YouTube, and it shows that the tricks played by the antibiotic. So this is a small one, edein, that doesn't let the messenger bind. That's all. Second one that you will see is tetracycline. It also works at the small subunit. It doesn't let the second tRNA bind to the second position. So one is bound, but the second cannot. Second position is called the A site, for those that want to know. So it just occupies it. Then I want to show two that work on the large subunit. First is erythromycin, which is one of the first that came into use in medicine right after penicillin. It sits in the tunnel. If you remember, the tunnel that protects the protein and doesn't let more than a very short protein to be made, and then it blocks. And the last one that you will see is disturbing the formation of the bond. Very clever chemically, maybe the most clever chemically. So here the bond is being made, and it just sits in the bond. No bond, no more protein. That's the whole story. So, sorry. All ribosomal antibiotics bind to ribosomal functional sites. We just saw it. But these are highly conserved because all ribosomes work the same way. I already said it. And mandatory for clinical use is the distinction between the patient and the pathogen. So how does this happen? This happens by subtle differences. Even in highly conserved regions, there are subtle differences. And I want to show you one to explain. So if we look into it, we look into the whole large subunit. This is the entrance to the tunnel, and erythromycin blocks most of it, enough not to let proteins go through it. So if we look at this position in the tunnel, like a perpendicular to its long axis, we see that there is a pocket, and the pocket is made just from ribosomal RNA, and all the members of the erythromycin family, which is called macrolides, are bound here. So some are bound excellently, some are bound very good, some are bound just good, but all of them block the tunnel. And all of them interact with one of the components of the tunnel that I marked as a pink, and it's adenine A in position 2058, according to E. coli. So this is correct for all bacteria. All eubacteria have A in this position. Let me show you how it binds erythromycin. So this is this A, and this is erythromycin, and the affinity here, the chemical affinity, is very, very good. It means this is very cleverly designed by bacteria. Now, please, ladies and gentlemen, pay attention to this point, and you will see the difference between bacteria and us. This is all bacteria and us. But when there is a G here, us, there is repulsion. This is too close here, the distance, and there is no binding. That's all between bacteria and us, between bacteria and any higher organism. Second question that I want to discuss, second point, is resistance. Resistance to antibiotics is one of the most severe problems in modern medicine. And let's talk about it. First of all, how it is being acquired. So I will show you one of the methods. Selectivity was shown here between this and this, and bacteria can just change themselves to G, and now they are resistant. Actually, resistance to antibiotics is a basic process for the survival of many microorganisms, regardless of their exposure to modern clinical treatment. And this is a very important finding. And this is one of the most severe problems, as I said before. The World Health Organization said we are reaching post-antibiotic era. It means the time that antibiotics will not be useful anymore. And even the World Bank estimated that up to 3.8, almost 4% of the global economy will be lost by 2050 because of resistance to antibiotics. Can we combat antibiotics fully? In my opinion, not. Because bacteria want to live. Maybe want is a street word for it, but this is what happens. And they are cleverer than us in terms of survival. But we can do some things. So our lab decided in our lab to help it, because companies are hardly interested now in antibiotics. Most of the big companies are not interested at all. So until recently, all the known structures were of models for pathogens, not real pathogens. And this explained the common modes of antibiotic action, as I showed previously, but they do not explain specificity of pathogens. And this is what we wanted to see. What's the difference between non-pathogenic and pathogenic? So we crystallized the large ribosomal subunit from a pathogen, actually a multi-resistant pathogen called Staph aureus. You can see here the crystals, and we got the structure. I won't show the whole structure. I will just show some lessons we studied by comparing it to non-pathogenic. So first of all, it's possible to increase antibiotic activity. More potent antibiotic can be made. We collaborated with a company called Nabriva, a small company in Vienna, and they made 16-fold stronger antibiotic. Lesson number 2 and 3 I skip. Lesson number 4 is beyond my expectation. novel antibiotic binding sites. I didn't expect it. It's a very, very nice surprise, and in my opinion, also very powerful. So if we look here in the structure of the large subunit in gray, this is the skeleton in two views. This little bluish, this little cyan, show where there are additions on the skeleton of the large subunit. You can see some on this side and some on that side. And maybe you can see even better here from all sides. We drew here only those that are on the surface, and those that exist only in Staph aureus, not in the pathogen models. So we suggest to use them as new positions for potential binding site, antibiotics potential binding site. And I want to show an example here. This is part of the ribosome. There are here two structures, one of the model and one of the pathogen. Here they are indistinguishable, but here there is a little addition. This is what you saw in this view. And we suggest that this can be potential binding sites because it's only in the pathogenic, and we found that it is also important for protein biosynthesis. So actually in a model system, we identified 25 new potential binding sites, unique, and 16 of them where they were blocked chemically, they inhibit protein biosynthesis. And because these sites are not involved in the primary ribosome activity, namely in the decoding and peptide bond formation, no pathogen contains genes for their modification. It means the bacteria did not understand now, although they are clever, did not understand now that they are important. So for now, they can be exploited for the design of specific advanced degradable antibiotics, hence environmental friendly. So I talked a little about specificity. I'll talk about it a little bit more, and about degradability. It means environmental concerns. So first, specificity. I hope that you all know about the microbiome. The microbiome is where trillions of bacteria reside in our body. They are called the good bacteria. They are usually in semi-exposed organs like guts, ears, lung, and skin, and they are mainly harmless. But they are susceptible to antibiotics. And so if antibiotics can penetrate into where they are, they can kill them too, and then people can catch other diseases. Therefore, we suggest to exploit these sites and design those that will not disturb the microbiome, the good. Also, they can be degradable. What do I mean in degradable? This is the environmental and ecology consideration. This is based on the fact that most of the ribosomal antibiotics that are used today are extensions of small organic inside the cores that are not digestible, and when they go out from the body with the rest, with the sewage, they are not degradable. And therefore, they can penetrate into the water. They are too small to be found by most of the used facilities for purification. And then they go into the water. They can penetrate into the irrigation water, and they come back to us. So this is why when we design our antibiotics, we want to make them degradable, biodegradable. So now I want a few words, how did we start? Because I thought that it has some lessons that we can learn from it. First of all, why did we use X-ray crystallography? Because human can see, I think, maybe insects with normal microscope cells, bacteria, viruses, but not atomic detail. For this, we need crystallography. For crystallography, we need crystals. They diffract. We see the diffraction here. They diffract according to the Fourier transform, and if we collect the whole three-dimensional, whole space around the crystal, we can get enough information to build back the structure. So here you can see X-ray coming. There is here a crystal. We collect the data with computers and with knowing how to do it. This little thing is a crystallographer. It means like something like me. And then we get the structure. So we can get structures of many types of molecules. But we need crystals. Let's define what crystals are. So this is sodium chloride. It's only 2 atoms, sodium and chloride. And it likes to make a 3-dimensional periodic system, even in nature, spontaneously. So you can see full periodicity in 3 dimensions of chlorine and sodium in between. This is a natural crystal. This is a crystal of a protein that was first crystallized in Oxford in the 60s, but a 6 years grader, about 12 years old girl, could repeat it. So proteins can be crystallized by this very complicated, now we know, very complicated structure that you know. But even before the structure was known, there was a big question mark. Actually, this question mark said no. And the people that tried to crystallize it, they explain it by all types of high degree of internal mobility, flexibility, structural heterogeneity, chemical complexity, all correct. But they paid attention less to a very important property of ribosomes, that they have marked tendency to deteriorate. Even when we prepared it, they deteriorate, and we get less than half of the expected activity. And I was much more open to this problem. So I thought, first of all, we have to find ways that the ribosomes won't be deteriorated when we made them. Okay, I thought about it, but I didn't know what to do until I read a paper about how is the metabolism of the sleeping bears in the North Pole. The North Pole, the bears are going to sleep for the winter, and a delegation went to see what happened inside them. So they found out that in every cell, inside the membrane of each cell, the ribosomes are fully organized periodically. Monolayers, not real crystals, but monolayers of organization. And this shows that ribosomes can be orderly packed. Every bear, every cell. So then I thought, why is it? Why did nature provide this mechanism? And in my opinion, this is the way to maintain a pool of active ribosomes for the end of the winter, when the bears would like to do other things and they need proteins to do them. So my conclusion was that stress induces periodic packing of ribosomal particles. So I thought, let's look at bacteria that grow under stressful conditions. I didn't want to work on the bears, on the ribosomes of the bears. First, I didn't want to kill more bears, and more important, because the ribosomes from bacteria can be useful for medicine, whereas those of bears not. So the solution was to use ribosomes from bacteria that grow under stress, like in the Dead Sea, in hot springs, or in waste of nuclear facilities. I just want to show you the Dead Sea. It's the lowest point in the world. It's in Israel. You see, from top, no animal, no tree, no nothing, no life. You can see from closer, these are all structures of salt that came out by evaporation. No animal, no plant. But there are 2 types of bacteria, and I want to show you the colonies of one of them. Heloarculamaris morti here, you can see it here too. And I knew about this bacteria, and we used its ribosomes. Also, we use ribosomes of Deinococcus radiodurans, which is a bacteria that can grow under super extreme conditions, very hot, very cold, environment, dust, no food, even under irradiation. And we got beautiful crystals. Look, how nice. So we got actually 25 types of them. I show here only 6, those that were useful for crystallography. And I want to show you 6 years after we started, we went to measure them. We took crystals to measure in this accelerator, which is in Grenoble. In Israel is a member of it. Actually, we used another one, but it's such a beautiful picture, I decided to use this. From top, it looks like that. Look how wonderful. From getting close, you see how it works. So here there are particles that be an accelerator, then they run around in this ring. And in the tangential, we have stations. Here there is a station that Frank is measuring on ribosomes. So each station has its own properties. Some are useful for ribosomes. So in 1986, 6 years after we started, we got the first diffraction. Look how beautiful, beautiful spots all the way to the end from the last subunit of haloarcula in time equals 0. We were really very happy, but not for long. Second picture was already deteriorated, damaged. It means the X-rays damaged the crystals. We repeated it 100 times, and we didn't get anything better. And we felt like we started from here, and we saw only this mountain. We call it the Everest, sorry. And we climbed it, and climbed it, and climbed it. Took 6 years until we got to the summit, and what did we see then? Not a really big, much bigger Everest, much taller Everest behind, that we will have to get there in order to measure. Much more difficult than until then. So this was the time I thought maybe we should stop this project, but instead I thought about why does it happen? All living tissues suffer from X-rays, but this is much too much. So I explained it by the crystals being weak or very soft, and the damage occurs in 2 steps. The primary is local disruption of the bond, and then when the bonds are being cut by the X-ray, the 2 parts of the bond are unhappy, going around and break more bonds in order to satisfy themselves, either to give an electron or to take an electron. And we thought that maybe the way to stop it was to cool the crystal so cold that there will not be energy for the running around. And this worked, although everybody didn't think so. Even I, look, this is the experiment done at Stanford. Look at my face. Even I didn't believe in our own experiment, but it worked beautifully. Have a look. And 20 years later, look how happy I am. Now it works, and this is the machine that cools, and this is the machine that measures the X-rays. This is in Grenoble. Actually, not only for us, it worked for the whole world. We introduced it here at the end of 86. Within the beginning of 87, 90% of the world used it, and now everybody. So what you see here is the number of structures that are known now and have been deposited in the protein data bank, and this is the year. And have a look, this is only till 2011. 47,000 structures. All of them done, or 99% of them done in our cryo method. And all of them are of very important materials for medicine or for making new materials. Actually, now it's 2017, and there are 80,000 new structures that couldn't be done otherwise. So how about future? We go back to the lecture, and what I want to tell you, we plan to identify the specific structural features which are unique for each pathogenic bacteria. What I showed you about Staph aureus, we want to do for all. Once we identify such features, we will design novel antibiotics for them. In these studies, in the novel antibiotics, we shall use cryo-electron microscopy, which provides high-resolution structures with no need of crystals. All what I told you until now about crystals, we will not need them in the future. Taking advantage of the current revolution in biostructure research. This is our program, and I hope that you support us. So before I finish, I want to thank the Weizmann Institute that let me work. Here is the president that made the decision, Michael Sella. He was a biologist. Although I was called the dreamer of the world, he still supported me. And when he finished his term, he was replaced by Chaim Harari, who is a physicist, and he also supported me. They were encouraged by the Scientific Advisory Committee, about whom I want to highlight Alex Rich, who tried to crystallize ribosomes with no success for 10 years, and he was still very supportive. Actually, the whole work started with a very strong collaboration with Max Planck in Berlin, with Dr. Wittmann, that he really wanted to see the structure of the ribosomes, and he helped in establishing a group for us in Hamburg near the synchrotron there. He died 10 years before the structures came, unfortunately, and then he was replaced by Franceschi and then by Fucini. So I want to thank my research group in the two places for their devotion and determination in good and in bad times for many years. Here, this is the group in Hamburg. It was terminated in 2004. It always had angels. The technical assistants say that otherwise they cannot work if there are not angels. Ribosomes are too complicated. And the group came to the Dead Sea to look for new bacteria. Well, they had good time. The group in Israel is still working. Here you can see people that came and went through the last 3 years. Some of them are still with us. I want to highlight Dr. Anat Bashan, who is the senior scientist that runs the show all the time when I'm doing other things. And I cannot show you one by one, but I want that you pay attention to Tamar. Tamar came 16 years ago for 10 weeks. She's still around. At the day that I took a picture, she had birthday, so she baked a cake. You want to see the cake? Here is the cake. And it shows that in my group, ribosomes are considered sweet. I also want to thank my family. I cannot show you all of them. My mother, my sister, and so on. Just my daughter, who is a doctor, a medical doctor, and her daughter. This is the girl that crystallized the ribosome when she was 12. And they were also supportive all the time, even when I wasn't there. My granddaughter actually gave me a speech and then a prize. I will show you the prize in a minute. In the speech, she said, as you know, she is a very busy scientist, but she always finds time for me. And this is the prize that she gave me. No, not this one. This is the medal, Nobel medal. Her prize is the Grandma of the Year is Ada Yonat, at least as important. And when I asked her, what is the year? Every prize has a year. She said, you have to reprove yourself every year. I'll take it off the wall if you fail. Still on the wall, you see? So ribosomes became popular. In a carnival, he is me. And she, his wife, is a ribosome. Look, protein comes out of her. Also, younger children became ribosomes. And this is what happened to me by one of the best artists and writers in Israel, Michel Kishka. And if you have a look very carefully, this is the large subunit. And this is the small one. And it's symmetrical. And he thinks that the best place for antibiotics is here and here. Thank you. Okay, I hope that you liked it. And I hope that something will come out of it, at least mentally, intellectually.
Yeah, Dr. Kakoli.
Thank you, Dr. Ada. Yeah. Hello. Yes, can you hear me?
Yes,
thank you, Dr. Thank you, Dr. Ada, for this invigorating talk. You amazingly explained such a complex topic in the layman's terms that is perhaps possible only for you. It was an honor for me to share your questions. Now we are open the platform for questions. Please type your names and I will speak out your name clearly. Are we having question and answer session?
Yeah, Dr. Kakuli, Madam wrote me, if there is any question, she will answer it through e-chat. So you can ask, but very limited numbers.
Yeah.
She will answer through e-chat.
Yeah. Sure. I just have one question. Can I type in or I will speak? What should I do?
You can
So,
ask for the question, and ma'am will
yeah,
ma'am will answer.
I have only one question. That is, you talked about specific advanced degradable antibiotics. I just wanted to know what is that degradable specifically means and what are their advantages. I just want that a simple question I have from my end. Hello.
Let's see, she might be writing it. Yeah.
Okay. And if anybody else has any question, you can just type in here. I can speak it out, spell it out for her. That will be easy that way. Is there somebody has the background microphone on? If you can turn it off because there is a lot of noise coming. Somebody's microphone is on. If you can kindly turn it off.
I think better to write the question in the chat box so that she can answer. She can write back.
Yeah.
Yes, Kakuli, the answer has come in the chat box. The
Okay,
answer,
I'll just
yeah,
check.
the answer is there in the chat box.
Okay. Thank you so much, Dr. Ada. Anybody else has any question? Any other question? Okay, we don't have any more questions. Let us thank Dr. Ada Yonath for this lucid presentation and sparing her time to be with us. We are really honored. Thank you.
Thank you, Dr. Kakuli Bose, for chairing this session. We find great delight in felicitation of our honorable speaker, Nobel laureate Dr. Ada E. Yonath. I humbly request Professor Pranab Ghosh, Department of Chemistry, University of North Bengal, and also the convener of Chemical Research Society of India, North Bengal Local Chapter, West Bengal, to felicitate our eminent speaker and say a few words.
Yeah. Good morning to all of you. Honorable Vice Chancellor, our Nobel laureate speaker, Professor Ada Yonath, Dr. Ajay Kumar Ray, and all the academicians, research scholars, and my beloved students from each and every corner of the country present in this meet. As the convener of CRSI North Bengal Local Chapter, I am really grateful to Professor Ada for her excellent and inspiring lecture on the topic Next Generation Antibiotics. For last 2-3 months, I was in touch with Madam, and all the time I received the positive response from her end. That was really enthusiasm, and it is also not for me, for anyone. Professor Ada, what I have learned from her could be a symbolic model for the young generation. Each aspect of her life was driven by bravery and determination. Not only she was an excellent student, but also a leader to her peers because of her high moral standards. The main motto of CRSI is to convey the knowledge of chemistry to everybody. In this context and during this pandemic situation, such an excellent lecture by Professor Ada, Nobel laureate 2009. What could be better than that? Yes, I do not know. The main motto of CRSI, as I mentioned, today's conference appeared as complete success in line with the objective of CRSI because we had a huge response starting from the school level students to the higher academicians. And finally, on behalf of CRSI North Bengal local chapter, I convey my heartfelt gratitude to Professor Ada. I wish her healthy and safe life to provide us with still new chemistry to provide. Thank you.
Thank you, sir, for your words. May I now cordially request Dr. Lizzie Matthew, Principal, St. Teresa College, Kerala, to felicitate our honorable speaker.
Dear respected guests, the Nobel laureate, Dr. The Nobel laureate, Dr. Ada E. Yonak, renowned scientist and professor of Weizmann Institute of Science, Israel, Dr. Ajay Kumar Ray, director, this Institute of Advanced Studies and Research, Dr. Sudhiresh Bhattacharya, Vice Chancellor of North Bengal University, Professor Anirban Mishra, Head Department of Chemistry, North Bengal University, Dr. Tapoli Bose, the Session Chair, Tata Memorial Center, New Mumbai, Dr. Rogi C. Joshua, Principal of CMS College, Kotte, Dr. J.R.T. Verki, Head Department of Chemistry, St. Teresa's College, Ernakulam, faculty members, research scholars, and students of chemistry and biology of various institutions attending from different parts of the world. A warm good morning to all. I feel extremely honored that St. Teresa's College could join the Department of Chemistry, University of North Bengal, and the JIS Institute of Advanced Studies and Research of Kolkata, and the Department of Chemistry, CMS College, Kerala, to jointly organize this international conference on Confluence of Chemistry and Biology, Exploring New Frontiers. I consider this as a great privilege to have with us Nobel laureate Professor Yonak. In this August conference, Professor Yonak had delivered an excellent lecture on new generation antibiotics. She started her talk on the structural basis of a fundamental primary metabolite, the proteins, the process of protein synthesis, that is the translating of the genetic code into proteins. She focused on ribosomes, the cellular factories performing their action and antibiotics. She revealed the novel routes for drug improvement and drug design in a very exemplary manner beyond words. She has also introduced several other important processes and terminologies related to ribosomal activity and the self-life process leading to the synthesis of new generation antibiotics. I feel greatly honored to felicitate this great resource person, the most prolific scientist, the first Israeli woman, Dr. Yonak, in this area of molecular biology, having won Nobel Prize specifically on the molecular structure of such a minute cell organelle, the ribosome. Dear Madam, we are indeed very fortunate for having your gracious presence, for having shared your innovative findings and contributions in the field of chemistry. It was an excellent presentation that you delivered, and of course, you have given us a deep insight during the course of your lecture. We are extremely fortunate for this golden opportunity to be amidst this illustrious crowd attending the lecture online with the first Israeli woman to win the Nobel Prize. Also, a laureate being the recipient of several other honors, and also the first woman in 45 years to win the Nobel Prize for Chemistry. I'm sure all the participants who have attended her lecture on this virtual platform have definitely benefited and greatly inspired and motivated and enriched by her lecture. I believe this international webinar has provided a broad platform of scientific interaction for the researchers, academicians, and and young students in an emerging area of chemistry-biology interface. I also acknowledge and congratulate the organizers of this international conference for their meticulous planning and every effort they have taken for the success of this webinar. My sincere thanks and great appreciation. Thank you all.
Thank you, ma'am. May I now request Dr. Varghese C. Joshua, Principal, CMS College, to kindly felicitate our honorable speaker.
Hello. Hello.
Yes, we
Am
can
I
hear
audible?
you. Yes,
I am
please, sir.
sorry. I am sorry. I am the head of the department, CMS College, Kottayam, Kerala. Our principal is not available now. Can I take the opportunity to facilitate from our side?
Yes, go ahead, sir.
Oh,
Briefly, you can.
okay.
You can.
We are very privileged to attend this international conference conducted by North Bengal University, Darjeeling, in association with this institute and Department of Chemistry, St. Teresa's College, Ernakulam. It is our uh, extreme thanks to the organizers to allow us to associate in this international conference, and also thanking Madam Ada to deliver a wonderful lecture to us. On the behalf of Principal CMS College Kottayam, I extend our heartful and sincere thanks to all the organizing committee and the Professor Ada Leonard to allow us jointly organize this function. Thank you.
Thank you so much, sir. May I now humbly request Dr. Prashanjit Shaha, JIS Institute of Advanced Studies and Research, and one of the convener of organizing committee for the deliverance of the vote of thanks. Please, sir.
Thank you, Devolina. Thank you very much. So, I represent JIS Institute of Advanced Studies and Research, uh, Center for Interdisciplinary Science in Kolkata. That is under JIS Group, the well-known group in Eastern India, educational group. So, when our director, Professor Ajay Kumar Ray, wrote a request email to Professor Radha, and she immediately agreed to deliver a talk for the huge number of researchers and Indian students for their benefit. And she immediately agreed and suggested that, along with North Bengal University and other colleges in India, can we organize a pan-India based lecture for the huge number of students? And from JIS, I would must say that we are proud that we can collaborate and we have organized this such nice event. So on behalf of the entire organizing committee, I would like to thank our distinguished guest, Professor Ada Yonath, for giving us her valuable time and for delivering this wonderful lecture. And that would be very, very helpful for all of us as the student of science, student of chemistry, and to cultivate this idea that we have got from her lecture that would make us inspired to do something in science. So thank you, Professor Ada. Although there is a minor technical glitch, but somehow we could manage it nicely. And yeah, I would hope that we would have you again in among us for such kind of lectures and interaction. And I would like to thank Honorable Vice Chancellor of North Bengal University, Professor Subiresh Bhattacharya, Honorable Director of JIS Institute of Advanced Studies and Research, Kolkata, Professor Ajay Kumar Ray, and all the dignitaries and our distinguished guests present here for your time. Thank you, Chairman, for this event, Professor Anirban Mishra, Head of the Department of Chemistry, North Bengal University. Thank you, sir. Thank you, Dr. Kakuli Bose, for chairing the session from Tata Memorial Center, Mumbai. Thank you very much, ma'am. And obviously, I would like to thank all the attendees and the participants, especially the young minds from all over the India for their active participation, and all the ladies and gentlemen who have participated in through Facebook or through YouTube or this event. And I'm sure that they will be benefited from this lecture a lot. And thank you all the colleges from the southern part of our country, Kerala, that CMS College and St. Teresa's College and their representatives, their principals. Thank you everyone. And I would like to thank my colleague Dr. Shubhankar Singho and Shankar Bhuiya for helping me a lot to organizing this event from our part. And last but not the least, I would like to thank my fellow conveners, uh, Dr. Shajol Das, Assistant Professor, Department of Chemistry, Dr. Rajesh Kumar Das, and obviously Professor Pranab Ghosh for being so kind and so cooperative throughout this event from its inception to execution. And once again, I want to state my humble thanks, and we are most grateful to you, Professor Yonath, for your time, for your lecture. And ladies and gentlemen, we thank you for being with us throughout this event. It has been a great experience. Thank you. Thank you so much. Yes, Jabolina, it's over to you.
Thank you, sir. May I now request Professor Anirban Mishra uh, to uh, to let us know that can I stop the meeting now or should I continue?
Yeah, now I think it's time. We had a very good time as we promised in the beginning that we will have a wonderful time. So we have had it. So with the permission from Professor Ajay Kumar Rai, we may close now.
Everybody for joining this. It was a great learning session for all of us across the country. And I'm sure that we will be able to again have such talks in future, and that too jointly. And we will get the opportunity to hear more from Professor Ada in the years to come. So thank you very much.
Thank you, everyone. Thank you.
Okay, Devolina, you can close it now.
Okay, sir. Thank you.